Assalamualaikum warahmatullahi wabarakatuh and good day everyone. Welcome to the IBD webinar series under the theme of Biotechnology, Science, Business and Society. I am Dr. Nuri Izian from the Institute of Bioproduct Development, University Technology Malaysia, and I will be your moderator for today's session. Thank you all for joining us. We are really excited for today's talk title, Roles of Analytical Scientists in Product Development, from cGMP analysis to permeation studies in cosmetics and pharmaceuticals. First and foremost, allow me to briefly introduce our speaker for today, Dr. Nazmul Alam. He is currently an analytical scientist with a strong background in both academia and the cosmetic pharmaceutical industries. He is currently a senior scientist in analytical development at DECIEM Incorporated, where he conducts thorough product investigations, performs data analysis according to cGMP and cGLP guidelines, and provides expertise in designing and implementing analytical methods. Prior to this, he served as a scientist at Dalriada Drug Discovery Incorporated, leading drug discovery projects and developing methods for quantifying drug concentrations in biological matrices. His expertise also include working as an LCMS scientist at Ditiba Incorporated, where he developed and validated LCMS-MS methods for analyzing specialized lipids and studied in vitro permeation of drug products for FDA submission. Dr. Alam holds a PhD in analytical chemistry from the University of Waterloo and has been recognized with the Right Elite 100 Award for Excellence in Project Management. He has a notable scientific impact with an H index of 18 and 1,194 citations. His research focuses on method development under cGMP standards, stability studies, and skin permeation testings, which play crucial roles in ensuring product safety, efficacy, and regulatory compliance. With his extensive experience, Dr. Nazmul has contributed significantly to bridging the gap between laboratory research and commercial product development. Without further ado, let's welcome Dr. Nazmul Alam to deliver his presentation. Dr. Nazmul, I hand over the mic to you and the virtual floor is now yours. Hi, Dr. Nazmul.
Excellent. Thank you so much. Let me just share my screen before I jump in. Can you just confirm if you can see my screen?
Yes, we can see it perfectly.
Perfect. So hello everyone and assalamu alaikum. It's a great pleasure to be here today to speak with the Institute of Bioproduct Development at UTM, virtually, of course. And thank you so much, Dr. Izian, for the kind introduction and for inviting me to this webinar. In this session, I'll briefly share my journey towards my current research role, where we focus on developing cosmetics products. I'll also be highlighting the crucial role in analytical department in overall product development process. So let's begin. Let's begin with the company that I work for. So now you see the company name. I'm just testing if you or the audience can answer my questions. I know we'll have chance for you to ask questions for me, but now I want to check if you can answer my question. How many people have heard about this company before? If you just say yes, I'll see how many people can recognize this name or understand what I'm talking about. So anybody who have heard DECIEM before, just say yes, then we'll see. Okay, we see answer coming out. Perfect. Yeah, some of you have heard of the name, but this is the company name. I'll tell you a secret why many people have not heard about this. Okay, before I begin, I ask the questions, how many people have heard? Let me give you the background. So DECIEM was founded in 2013 with a very clear mission to fundamentally change the beauty industry by challenging existing norm at that time. So our founder, Brandon, envisioned a world where skincare was built on three things. One is integrity, innovation, and transparency. And also is free from the marketing fluff that too often we see, and those marketing terms overshadows the real science. So our founder decided to call the company DECIEM, followed by the abnormal beauty company. This is the tag. Their goal was to shake up the industry by doing things differently, making a skincare accessible, transparent, and developing product with integrity. So that's why we named our product after the ingredients inside them. Probably you have seen some of the ingredients name as a brand name. This continues to be really important value for us. Now, probably you have seen some of these names: The Ordinary, Neod, Loofah, Avestin. So I'll briefly talk about these four brand names that DECIEM produces. So the first one is Ordinary. This one all about being clear and straightforward. It gives you good science-backed ingredients without breaking your bank account. And the Neod, if you're into the newest skincare innovations, for example, and this is for you. It's our most advanced brand, using cutting-edge stuff, really optimizing your skin. And then third is Avestin. So Avestin, this is completely different. This has also emotional connection with our founder. There's only a store in London, Ontario, not Ontario, London, UK, that sells Avestin. So this is the rethinking the fragrance, so moving away from traditional perfumes to create scents and that are unique and also feel a bit familiar. And the last is the Loofah that I want to just briefly talk about. The Loofah is our kind of newest brand. It's also rooted in scents and self-care, but with scientific touch. So right now, we have scented body wash, also paper ink, and more products are coming soon on this brand. Just to give you a few examples, pictures, as I said, the four different main groups. I think Ordinary is the most famous, especially in APEC countries or Asian countries. That's what I have seen. Although, as I asked you before about the DECIEM, many people have not heard about DECIEM, but I have observed that many people have heard about Ordinary. And when I show the image or picture, they can recognize it. Anyway, so since 2013 in Toronto, the development, now DECIEM has been creating innovative products and formulations that is helping people all over the world. But the question is, how do we make this formulation perfect that people loves? So I'm going to give you a little bit of behind the scene work that we do. Some of the steps that you are probably interested in. So among our audience today, so maybe some of you are entrepreneur who wants to develop cosmetics products for future clients. Some of you are probably developing ingredients or base or supporting materials that will eventually lead to some formulations. So the materials, the slides I'm presenting next will be useful to understand the process. And if you're a student, for example, if you want to work in cosmetics or pharma industry, it will be useful for you to understand how industry develop a product from start to end. So I know we don't have so much time, but I'll try to briefly explain what we do at DECIEM to make a product. Again, the same process can be translated into different industries. That's why I made it a little bit of generic. I know this is a busy slide. The step-wise, the first step is the concept and prototyping. So this is kind of idea spark stage. We do deep ingredient research. We figure out the exact skin pathway we need to hit. Then we immediately start formulating prototyping. Sorry, formulation prototyping. We test science right away. Okay. The early focus saves every time. So this kind of first stage. The second stage is the analytical. This is our quality check. It's a kind of iterative process that pushes product development forward. We use equipment to confirm purity. We verify the precise concentration of every active ingredient. No guesswork, just hard work. And the third one is stability. This is our super important. We put formula through a stress test like heat, cold, light. We confirm it won't separate or degrade. This links the formulation and analytical development is very, very strong. So we work very close together, and we promise the product works just as well as a year from now as it does today. Huh? Okay, so then microbiological test or microbiological department. We have separate department. Microbiological is about safety. We test our product to ensure it's clean. We check for any harmful bacteria or mold. This step literally lowers waste. It makes the whole production process smoother and more efficient. Safety is non-negotiable for us. You know. Now the last stage in the scientific development is clinical, the final stage. This is where we prove it works. We conduct human trial with objective measurements. The goal is clear: to validate every claim that we make. This data helps everyone focus. More importantly, it gives you confidence in the proven efficacy of the product that we claim. So this kind of over 5 stages. I just briefly mentioned 5 stages, and here might be more steps, but I just generalize it into 5 stages that we follow for our scientific development. Now, where as an analytical development we fit in, I want to highlight a little bit more or expand the analytical development role. So analytical development is the center half of our 4 functions in our business. Excuse me. It's not just testing, it's about strategy, compliance, and growth. The first one being the method development. This is fundamental. We must create or adapt testing methods based on product type and also sometimes instruments available. We need to know how to accurately measure a product before we can measure it at all, right? It ensures our results are reliable, and every time it's reliable. The second function that analytical team does is the label claims. So the analytical team is essentially for supporting every claim we make on our bottle or box. So why label claim is important? You see, the DECIEM produces cosmetics that are a little bit different. So we produce functional cosmetics. So always you will see as a 5% this, 3% this, something like that. So you have to substantiate that whatever label number you're putting is really scientifically defended. Okay, so our lab provide the hard data and the evidence that needed to legally and accurately state that the product has, for example, 5% active ingredients. We also do investigation and compliance. So why do we need that? This is crucial for safety and product life cycle or regulatory support. So if there is an issue with a batch, a team investigates the root cause, and they ensure we always meet global standards, keeping our product on the market and consumer safe. As you probably know that market leads the product criteria or specifications. For example, if the cosmetics product is for China, it has to follow certain specifications. So we develop, we test for that particular market too. We also work on special projects. Um, so we have a special projects in analytical team that supports all growth initiatives. For example, we test new market expansion. We also manage data for reformulations or addressing any unique that on-off testing needs that we support also. So these four roles ensure analytical development is an integrated part of the whole product development lifecycle. Okay, so this is kind of brief. I'll keep more information for the question answer session. If you have some question or you want me to explain specific topics, definitely we'll have a chance at the end. Okay, a little bit shifting to my journey. I think that will help to understand how a scientist work in an industry. So education-wise, I studied multiple places. The first being BSc and MSc in chemistry in Bangladesh. And then I moved to Japan. I did a diploma there. And then I moved to London, Ontario, Canada. I did master's again. And I moved to another city called Waterloo for PhD. During this period, I studied different topics, like starting from synthesis to biomaterial extractions, biosensor development, and also analytical chemistry. So I wanted to highlight one specific topics that is very close to my heart and help me what I do now. So this is called solid phase microextraction. I was working with Dr. Janusz Poglishin. So this is a device he invented, and now it's being used in different applications. I'll show you in the next slides. So what it does is it's a device that extracts active ingredients from your sample. So sample could be different things. This is a simple schematic diagram. And then once this fiber coating or the device extracts enough analyte from your sample, you can either dissolve into a small pure solvent and inject or detect what you extracted, or you can directly inject this to your GCMS, for example, instrument. So this has multiple applications already, including tissue, urine, and different mode of applications. Probably some of you have already heard. So these sample preparation techniques is close to SPE or solid phase extraction. Is more of micro extraction, but this has other features. So why I'm saying this is in my PhD, I not only did the experimentation with this device, but also I did mathematical modeling to understand how this device works. So I parallelly worked with experimental settings and also model. So just to give you one example how this model helps. So as I showed you the SPME fiber, so how analytes diffuse and extract and deplete around the fiber, that is shown in this image, and that is mathematical model using COMSOL multiphysics. So now, why I'm showing this? I'll connect this my education towards the work. So from my education to work at different places. So I studied, I taught at a university in an early career, and then worked at multiple places and worked with different applications and different laboratory settings. Now, this helps me to understand the science behind sample extraction, how analytes migrates, how a drug molecule retain in one phase and moves to another phase. So this experience that I gained from multiple places is useful for a scientist to investigate a product, to plan the next stage in our product lifecycle. Now, also this gives me exposure to different instrumentation, different softwares that helps me to design a protocol that is necessary for a challenging project. Also, throughout my journey, as I worked with different industries and different labs. We have learned pharmaceutical R&D and drug discovery, PK studies, bioanalysis studies, and natural health products. So again, why I'm showing this as a scientist, uh, this is kind of typical journey where most of the North American scientists, as far as I know, they went through. So if you're a student, if you're early career interested in working at industry, so this is how you can envision yourself that gaining multiple experiences will help you to establish a position in the industry where you can solve problems. Okay, let me shift gear a little bit to a specific area that I want to highlight a little bit. If you are, let me check my share. There's no message for me. Okay, good. So in this slide, next couple of slides, what I want to do, I want to deep dive into a test that we do for cosmetic product development, especially when your cosmetic product is not just a cosmetic, it's like functional ingredients that does something with your skin. So we have to do these studies. Now, what you see here is the skin. If you put a drug or your cosmetic that has active ingredients on the skin, what it does is it can penetrate through your outer layer to even your bloodstream, depending on the nature of the active ingredients. So we have to, as a product development scientist, we have to understand what ingredients we add to our formulation and how far they can penetrate, because that's the safety and efficacy issue. Now, how do we test this? There are multiple ways. One being in vitro method, that where we use diffusion cell, other being ex vivo or trapping. In vivo method is more of pharmaceuticals. In cosmetics, rarely we use it. So I'll highlight this process a little bit. Okay. Yeah. So if you see on the left side of your screen, so that's the setup, experimental setup. It's called Franz cell. In this cell, you have at the bottom cell, you have a reservoir or solvents. And the top cell, you put your drug or formulation. In the middle, where you have a membrane. So you put your membrane formulation on the membrane and allow formulation or drug penetrate through the membrane and reach to the solvent or the sometimes solution that you use as a reservoir. And then we collect sample at a specific times. So these samples then processed and injected or quantified with instrument. So what it gives is a time profile of the actives that penetrates through the membrane. Now, why I'm saying membrane? This membrane could be a synthetic membrane or could be real human skin or some models. There are other models too. So the goal here is to check what's the time profile of penetration of our active. Now, this setup also helps formulation team to optimize the product development. For example, you have 3 formulations prototype, and we want a specific penetration. We want a specific time profile. Then we try 3 different formulations and check which formulation works better. This one is better than doing it all together in a vessel. I have seen the people do is everything mixed together in a centrifuge tube with the solvent and then extract. So that way we don't see the real picture of penetration through a layer. So right now with this experimental setup, we can clearly identify that which formulation works better, and also we can extrapolate this information towards the safety and efficacy. We can definitely talk more about these if you have questions. So what we have discussed so far. a few things I want to highlight is a typical journey of a scientist. So if you are a student or young scientist, then you might now understand where the journey could be for you to work in an industry. Or if you want to switch from academia after PhD, postdoc, then going to industry and working in the industry settings. I tried to highlight this point. And then I tried to highlight a few studies that we do in skin care product development. One being the skin penetration studies, which we often do for ingredients that we suspect that they can penetrate through the skin and reach to the bloodstream. For regulatory reason, we also need to show these data sometimes. Again, it depends on the market and product type. So I think I have give you enough to continue conversation, and I'd love to hear more from you, and so that we can continue our discussion. Uh, Thank you so much for your attention. I know listening to someone for half an hour, 40 minutes, 30 minutes is not easy. So thank you so much again. And if you want to connect me through my LinkedIn, here's the QR code. And thank you again. I'll be giving Dr. Islan for the question sessions. Thank you.
Hello? Is it
It's good
can you
to
hear me?
hear now.
Oh,
Yeah.
okay, sorry for that technical error. Okay, thank you very much, Dr. Nazmul, for that insightful and comprehensive presentation. We will now move on to the Q&A session. For participants, you may type questions in the chat box, and I'll try to read out a few questions to get us started. Okay, while waiting, Dr. Nazmul, I would like to ask several questions on my side. Okay, as you have presented in your presentation slides just now, you have mentioned about the five critical stages, whereby it consists of the starting with the concept and prototype, and then goes to the analytical methods, and then the stability testing, microbiological, and finally the clinical testing. Okay, so my question is, based on your experience, which stage is the most challenging and the most expensive in terms of the cost? I guess in terms of the cost, maybe the clinical trial or the clinical part will be the most expensive. I'm not sure, based on your experience in the industry side. Uh, but what about the most challenging part among these five stages?
Very good question, Dr. Zian. But it's very difficult to answer which stage is the most expensive. Now, I know in industry, money is the first thing that we consider. If it is
Yeah.
cheap, then it's that's very important questions. Now, it depends on the product type, and it depends on which area you want to target. The area, I mean, is if it is skin, or it is one area, and also the area, I mean, also the market. So if you go for, say, China market or European market, then we have to follow different guidelines. So the
I
expense
see.
also piles up for that. Now,
Okay.
in general, concept, prototype, ingredient starts, all are kind of desk work. The moment you have to work on the bench, your expense goes up. So analytical sometimes considered as expensive because of, we we put our money in the drain essentially. We just test and this goes.
Mhm. Yeah.
And but it's crucial because analytical and stability, if we don't do this well, then
Mm.
we cannot substantiate our label claim.
Mm.
We, even if it's clinically efficacious, you can demonstrate that okay, you have clinical benefit out of it, but we have to substantiate that based on analytical stability. So overall, since our products are different from many cosmetic products, so our products are like functional cosmetics or
Mhm.
cosmeceuticals. So we want active ingredients. Our labels, most of our products has label claims. So that's why we cannot bypass these expensive steps. So we
I
have to
see.
follow analytical, we have to follow stability, we have to follow
I see.
microbiological, and then clinical. I think all cosmetics product or companies, they do clinical. I think that's their main goal because
Mhm.
that's what they can market, right? That's what people see, clinical efficacy.
Yes. Exactly.
But for us to reach to that point, we have to go through multiple stages. Although they're expensive, we have to do it because of the functional nature, the nature of the business.
I see. So, so for that summary, it means that for all types of products developed by DCM, it is compulsory to undergo all these five stages without skipping any of the stage, isn't it? So,
Right.
regardless of what type of products it produces by the company, it has to go all to these five stages.
Right, right. And yeah, so sometimes these stages can be very thorough for some products.
Uh,
For some
Depending on.
products, maybe shallow. Um, Depending on the source of the material. So if it is
Oh,
chemically made, say chemical like retinol, if it is niacinamide, we already know their products, so byproducts, and um, this is kind of controlled molecule where
I
you
see.
don't need, sometimes you don't need thorough investigation in your formulation, but some products are like peptide based natural extracts, but we also claim certain percentage of those natural extracts. So in that case, quantification, identification, and also microbiological biological testing sometimes challenging, and
Uh,
those are expensive to
yeah,
substantiate that. Yeah, so it
yeah.
depends on type.
Understood. Okay, now I can see that there are several questions by our participants. Maybe we can pop up the first question. Okay, Dr. Nazmul, we have one question. The first question by Rahimah Sabtu. Her question is, how do you correlate analytical data such as particle size, viscosity, active content with product performance and sensory attributes?
Yeah, very interesting questions. Thank you so much, Rahima. Um, we have multiple teams. Yes, we have different instruments to collect all this data, and then we combine those data to support the formulation team. Okay, so
Mm-hmm.
it's not one person job. So particle size and viscosity is done by one person, for example, in analytical team, and then active content in another team. So the question is here how that correlates to
Mm,
my performance. So
yeah.
it depends on your product. Say if it is emulsion, then your test type will be different than if it is a cream or the serum. So number one is the number of tests or the type of test depends on what is your product and what we want to claim at the end. Now, how this correlates based on the final expectation or we call COA, like certificate of analysis.
Uh-huh.
We can go back. So, what certificate of analysis? What's the range? The criteria we should be within, and then we test that in our lab, and then see if we can achieve that. Our formulation is achieving that spec or not. So it's again it's a reverse engineering. So we have a spec. We want our, say, particle size, we want our viscosity
Mm.
within a
Mm.
certain number, and then
Yeah.
we test. So if our test passes, then only you can say, hey, we have good performance, or we have good sensory data. Uh, so that's how I think I was able to answer the question. So how you correlate data, meaning we don't have to correlate data as per se, but we have to um, have the data within our spec.
Yeah. Okay, I hope that's answering a question by Rahima. It means that normally what DCM or any cosmetic based industry did is normally they have their final target for their respective product, and they will try to work on it to achieve that particular target. If still not achieving that target, so it will go back to the formulation and analysis and towards achieving that particular target, isn't it?
Yes, yes, you said right. Yes. So that particular target is the first stage, as you see here, concept and prototype. So during that stage, we understand about the formulation, our expectation, right? So we want a product that will have certain criteria, that will have certain pH, certain particle size, certain thickness, density, etc. Right? So we a formulation team tries to achieve that, and analytical team um, uh, test that in every stages. And
Mm.
sometimes we have, for example, we have say 6 different formulations prototype,
Mhm.
and we are testing, and we saw one formulation is say color changing or degrading or the phase separating. Now, but that has a good clinical efficacy, or that has a good uh, possibility for final product, but the phase is
Mm.
problem or pH is problem. Then what we
Mm.
do as an analytical team, we investigate.
Mm.
We check what causes this, and then
Mm.
we look into all the ingredients. We do all our investigations. We use different tools. Well, sometimes we use mass spec. Sometimes we use NMR. Sometimes we collaborate with other department, university, or industry to
Mm.
understand that formulations, why this is going on. And by this process, we understand the formulation, we understand the problems, and we solve the problems. So sometimes it takes longer time for those type of formulations if we have these
Um,
challenges.
yeah, okay, I understood. Okay, Dr. Nazmul, I'm chipping another question. Regarding the target, the end target, is it normally the target is being made by the company itself, or is it being made by the customer, the final target in terms of the criteria for that particular product?
What I'm reading your question is how we select a product that we want to make.
No, no. I mean that normally the target for a particular product, for example, in terms of you want it to be in a certain pH, in a certain particle size, for example, or is it in a certain texture, for example, or color, is that been set by DCM itself, or is it or is there any prior survey been done by the customer to have to get to know the criteria that most of the customer or the clients outside there wanted that product to be?
Oh, I got it. Okay, so it's
Yeah.
a multi-step process. A multi-step process. Yes, it's done by 2 teams. We have 2 teams: early stage research, and then we have another team who does the market research, and also we have brand who
Mm,
into the branding, and then they understand the client's need, and
mm,
also they try to understand the ingredients functionalities.
mm,
So based on that understanding, we our formulation team then prototypes. So before even reaching to the COA level, we have 3 teams. They work together to write a certificate, COA certificate
uh,
of
Certificate of
analysis.
analysis.
Right. So that's where we're talking about, right? So to get that certificate of analysis, it's not just from a vendor, because our vendors are raw material suppliers.
Mm.
They are expert in their raw materials, but we make our formulations.
Mm.
So formulations are based on, as I said, three different teams who work together, who does initial research, and then this research, both in scientific research and also market research, brand research, and then formulation team, they start working on it. And I remember we have hundreds of prototypes for one product.
I
Um,
see. Quite a
not just
huge,
Yeah,
quite a huge number. Oh, okay.
yeah,
I see.
sometimes it's really big number.
Mm. Okay, good.
It's to track all these numbers. Yeah.
Yeah. Okay, Dr. Nazmo, it seems like we have another question by Efa Liza Misran. Good morning.
Yeah.
Good morning, or maybe good evening. In Toronto. Okay, she has a question. What strategies can be applied to ensure data integrity and traceability under the cGMP for complex product matrices?
Um, this is very, I think, very detailed questions. There are so many pieces I can read here. So data integrity, traceability. This is one thing I can summarize, and then cGMP. This is another, and then complex product matrix. This is another. Okay. So data integrity and traceability. Uh, in cosmetics, we are not very good. I mean, whole cosmetic industry are not perfect role model for data integrity and traceability. That's, I should say. But we have role model to follow. Pharmaceuticals, they have developed already cGMP for years, and as the question said, what strategies? So we don't have to invent our strategies as a cosmetics industry. We can just borrow the strategy from pharmaceutical industry that they have
Uh,
already established
I see.
cGMP. Okay.
Mm.
Now, this one part. The other part is complex product matrices. Yeah. So formulations, the cosmetics is really complex product matrices. Like in
Yeah.
pharmaceuticals, we do bioanalysis or DMPK analysis. Our matrices is tissue or blood. Yes, they are complex. Also, cosmetics products are complex. We have lipids. We have
Mm.
wide varieties of matrices.
Mm.
Now, these complex matrices analysis needs method, and method needs to be robust. And that method development and validation, we follow some GMP guidelines. And also we have cosmetics guidelines, I think 95106, I forgot the number. So we have
Mm.
cosmetics guidelines too.
Mm-hmm.
But for cGMP, we have products, not only cosmetics, we have also OTC products over the counter. For OTC products, we have to follow cGMP. But for some cosmetics products, we our cGMP is kind of not up to the level of the pharmaceutical cGMP. So that's what we as a, if you're thinking of developing your lab or setting up your company in cosmetics
Mm-hmm.
area, so I wouldn't scare you by giving this cGMP and all this data integrity. For cosmetics, it depends on what type of product you have. If it is OTC product, you are giving level claim, you need a little bit of stringent lab conditions, more data integrated things. But
Mm-hmm.
if you're just cosmetics area, maybe you don't have to worry about too much about cGMP.
I
Yeah.
see. So, the cosmetic regulation and the requirement is much more less stringent compared to the
Yes.
pharmaceutical.
Yes.
Yeah.
Yes.
Ah, okay. Because in your presentation, you've mentioned quite several times regarding the China regulation. So,
Mhm.
which region have the most stringent requirement in terms of the label claim? Is it China?
Um, I worked with different products. I saw different products as different criteria. So our regulatory team evaluates those criteria. So we have Japan, we have
Mm,
registration for Japan, we have
mm,
China, and we
mm,
have European Union. So Europe
mm,
also a little bit very cautious on safety and
Mm.
efficacy things.
Yeah.
In the states, we have regulatory based on different states. It's a California. It's different
Mm.
than it's in New York. So some of the states are relaxed, some of them are very tight.
Yeah.
So then again, to answer your question, it is not very general that always China, but yes, China is one of the tightest regulations they apply.
Mm.
Also, Japan, they also apply very tight regulations. Yeah.
Ah. Okay. Okay, moving on to the next question. Okay, the third question by Muhammad Amirul. Good morning, doctor. Good evening, Dr. Nazmul. What does cGMP mean and how does it affect the quality of what we use every day? Is it compulsory to follow the cGMP always for every product?
Got it. Got it. Okay, I see. There's interest on cGMP, this word or this acronym. So,
Mm-hmm.
good manufacturing process. This is very broad term, very broad term. Now, this term is usually applied for pharmaceuticals. And pharmaceuticals, uh, if I can give you a little picture on pharmaceutical developments, for example, the tablet we eat, say Tylenol or paracetamol. I don't know
Mm.
how you call it in Malaysia. We
The,
call
the
Tylenol paracetamol. Okay.
yes.
Um, so the tablet you take, how long? Let's imagine this is a new product, new tablet. It takes 4 or 5 years to go to the market from start, right? How?
Yes.
There's so many tests that has to be followed because this tablet goes through your body, goes to bloodstream, kidney, and all this metabolism that goes through.
Mm-hmm.
So GMP is applied for those development because we are taking this and we are using these ingredients to do something in our body. Okay,
Mm-hmm.
but cosmetics here, some of the cosmetics they work only on the surface, so
Mm-hmm.
they don't penetrate, they don't have any active ingredients that really interact with your metabolism. Some of them.
Oh.
So for cosmetics, you don't have to follow the GMP because uh, to sell in the market, what I have seen is the authority, that market authority, the regulatory body, they don't have strict rule as they have for pharmaceuticals. So
Ah.
as an inventor or as a company, you don't have to follow CGMP for all cosmetics based on the regulations, based on the market. Now, when do you need CGMP? You need two things. To credibility, like as a good company, you show to your clients or
Mm-hmm.
your integrity, you show your integrity that what we are doing, what we're testing is our GMP environment. Like DECIEM, we like when DECIEM started, they didn't have GMP in the earliest stage, like a few days,
Mm.
couple
Mm.
of years, and then they implemented all GMP. Now we have GMP systems in place.
Mm.
um, so what I'm giving two messages. One is yes, GMP is important
Mm-hmm.
uh, at different levels. So if you're starting out, then I wouldn't scare you about this GMP and spending money on GMP things.
Mm-hmm.
But if you want to be a reputed or if you want to show integrity that my product is really safe and efficacy is good and clinically it's proven and I don't have any harmful stuff that causes your damage and things like that, then
Mm-hmm.
following this test in a GMP environment helps. So GMP is like controlling your test and environment. Is that you have to have SOP, you have to
Mm.
have people trained, you have to have an instrument or system is qualified. So
Mm.
whatever data you are generating, you
Mm.
can trust, you can defend. So that's essentially GMP means
Yeah.
um, in a nutshell.
It's
Yeah.
it's like the SOP that the factory or the manufacturer needs to follow. In general, the GMP, the good manufacturing practice, uh, but for the cGMP, it is another level on top of the GMP.
Yeah, it's a recent one. It's a current GMP.
Mm,
It's kind of recent. GMP started with GMP, and then now it's a
mm,
current GMP. It's a very big term, and so many people are involved with that.
mm,
So many things. But in for us, what
mm.
we need to know is essentially it's a structure, a framework. I would say it's a framework um, that guides an industry to do each stages in a systematic way.
Yeah, systematic
So
way.
if I follow GMP in Canada and in Malaysia,
Yeah,
it's exactly the same thing. We can expect that, hey, the results are similar because we are
yeah,
following GMP. So it's kind of guideline.
yeah. Yeah, and this is also important in terms of the audit, something that needs to be carried out in at the industry. They have I believe that DCM will also have several
Yes.
audits. So this by having this GMP and cGMP, it may ease in terms of the documentation for the audit. Yeah.
Yeah, yeah, we have to have. So
Mm.
if you claim that you are GMP certified, so how can you claim? So you have to be audited by a company or by an organization.
Yeah.
And then if the audit, they will have a checklist. If
Mm.
you have this, okay, check this,
Yeah.
check.
Mm.
If not, you're not certified.
Yeah.
That's it.
That requires lots of work and documentation and
Right.
the involvement of all the staff there.
Yes, I
Okay.
know. I know why. I know if you're in academia,
Mm,
this term is vague. I had similar problem. Like before coming to
mm,
industry, I see for job applications they said GMP experience. Do you know about GMP? Things like that. Okay, before going to industry, I didn't have also clear understanding of GMP. So if our audience is student or academia, for you, GMP is, as I said, it's a big term, but nothing but a framework that every stage you do in your lab, for example, you come in, you have to follow a rules or guidelines. So you come
mm,
in, the guidelines, your study
yeah,
work guidelines. So you follow guidelines. So essentially, you are not you're using brain all the time. You are using guidelines. So say if you have a problem with the instrument, follow a guideline. Meaning,
yeah,
you cannot just yeah use your instinct. Okay, I know how to fix it. Okay, let me fix it and rerun the sample. No, you cannot do it
yeah.
because there is a protocols. If you have your run or batch fails,
Mm,
so you have to follow the SOP. Okay, if it fails, then stop. Don't do it, anything. Talk to your
mm,
supervisor. So those guidelines or the steps that are
mm,
very strict, that are
mm.
written, documented. As a chemist, analyst, scientist, we have to abide by those. That's essentially GMP.
Yeah. Okay, a great great and clear explanation. I think that's answering Muhammad Amirul's question. Maybe we can have one or two more question. Okay, by another question by Naja, baby food. Maybe from our industry partner, probably. In your opinion, Dr. Nazmul, what is the next frontier for analytical science in the era of personalized and biotech-based cosmetics?
Wow, it's a very academic questions. I'm trying to understand the next frontier for analytical science, personalized and biotech-based cosmetics. Wow, so it's a huge personalized medicine or personalized cosmetics is a buzz and hot topic now, and also biotech based. It's also booming. People want more natural, more sustainable products, and that can be done from biotech based. And then personalized now because of the AI, we can generate our personalized the solution. For example, in cosmetics field, if you scan with the robot your face, they will recommend your solution, something like that. Now also people are using AI to formulate based on that person's profile, formulate a formulations. I see there is a product where you have dispensers, two, three dispensers, and based on your need, you switch buttons and you make your own formulations from the dispenser. So these are kind of personalized medicine going on. Now, for analytical science, there is a challenge. There are two things: personalized medicine challenge and biotech challenge. For biotech challenge, is not new anymore because it's the same challenge as pharmaceuticals. So again, I can correlate this very well because I worked in pharmaceutical industry before cosmetics industry. So, the problem we have in pharmaceutical uh, now we're going to have this problem in cosmetics. Problem, I mean challenges. What I mean is analytical challenges. So now we see more biotech-based cosmetics. We see those challenges that pharmaceutical industry have already seen before. So what I'm saying is for analytical science in cosmetics, these are kind of not new. If we have experience from, or if we can borrow knowledge from pharmaceutical, we can just apply to cosmetics also. So that's the challenge. And then for personalized, again, cosmetics is unique for personalized. I know in pharmaceuticals, personalized medicine is a long way to go because of those regulations. But for cosmetics, people have already started using personalized cosmetics. Companies are offering different personalized solutions because you don't need to go through strong regulations. Now, for analytical science, personalized medicine, it depends on the product to me. If you, again, don't claim ingredients, if you don't have to abide by some regulations, then analytical science is not expensive anymore. You are not doing anything extra. But if you want to do something close to OTC, then definitely you have more cost uh, on each stages. I hope this explains. I think this is very general or broad questions. If you have more specific, let me know, or we can talk more.
Okay, I hope that's answering Naja Baby Food's question. Quite interesting and very in the future perspective. That kind of question. Okay, maybe we can have one more last question by our participants for today's webinar. All right. This may be our last question before I wrap up by Farhana Hasmuni. Doctor, how can analytical data support product claims such as long lasting, fast absorbing, or clinically tested? Thank you.
Interesting. So it's a label claim people make on the product, right? So it's a long lasting, it's fast absorbing. Okay, so let me add this one by one. Long lasting. What does it mean? What does it mean by long lasting, right? So long lasting could be your product that you apply. It um, it can retain for longer. Dr. Isyan, let me, if you have different understanding, let me know. So long lasting meaning you're applying a product, you can retain, or you don't have to wash your face, say, next 8 hours. Is that long lasting means?
Yeah, probably no need for frequent reapply on the
Yes.
skin,
So
something
it's
like
kind
that.
of in pharmaceutical we call doses. So how often it is? So paracetamol, Tylenol every 4 hours, right? For antibiotics every 8 hours. So for long lasting means your ingredients. Think similarly. Why do you need to take um, paracetamol or Tylenol 4 times, right, or 6 times a day? On the other hand, some medications just once in a day. Now, which one is better? Once in a day, very definitely better. You don't need. So same is the long lasting. Now, why? How do they make the formulations? So the formulations retain your active for longer time, so it doesn't degrade. Now, it all about the formulations, how you formulate your active ingredients. Now, question is how do you support this? For long lasting, we do stability experiment. We do a stability on in vitro test level, clinically. Before clinically, we do also in the lab, analytical lab. We have different setup to test. Also, we have shelf life study. If it is long lasting, meaning I can buy one product and I can use for 2 years, for example. So we have stability data. We have to show that okay, this product uh, you can use for 2 years, 3 years, no problem. Okay, that's long lasting. Now, fast absorbing is again, I showed you the skin picture. Absorbing means you apply and you're active being absorbed by your skin. So absorbing essentially is just a general term. Sometimes we the people use term that client understand, but it's absorbing essentially. It's diffusing through your skin and like penetrating and interacting with your internal organs. Now, fast absorbing, what the selling word is like. So you want a treatment, right? You want a treatment, so you apply, you get a treatment, like aspirin. I'd say, if you have stomach pain or you have pain, take aspirin. Within like, quick, you relieve. So that's the selling feature of aspirin. Why people want to take aspirin even if there is a side effect. So fast absorbing is that. Clinically tested, yeah, it's obvious that our product is clinically tested, meaning this product was applied to people, like in clinic. So people can, and on a regular basis, say if we have to claim that if you apply 7 days, you will see difference or you see improvement. So we want to test this clinically, meaning we send this product to people, or volunteers come to our lab, our clinical team lab, and then they apply in a certain condition, say half phase, sometimes half phase, different placebo, and then the volunteers are being tested for a certain period of time. Um, and then those results are compiled, and then those results are submitted to the regulatory as a support analytical data. I mean, these are analytical data to support for the label claim.
Okay. Just a little bit more addition in for this fast absorbing. Is it always correlated with the size of the particle for the in the micron size, or if let's say the product is having a nano sized particle or emulsion, so it means it has a faster absorption compared to the micron or non-micron one?
Um, Actually, no. Let me just show you the skin. Where is the skin? Let me show that way we can better understanding. Yeah. So if you see the skin, you have multiple layers. The top layer is very rigid, stratum, and then when you apply your cosmetics, you see the cosmetic formulation has different particle size, as you mentioned. The Typically the particles are the vehicles that are supporting your active ingredients. Now, this fast absorbing tells you the absorption of your active ingredients. Those are molecules. So we wouldn't consider this as a particle. Rather, the nature of the molecule. If the molecule is lipophilic or hydrophilic, and depending on that nature, they can absorb or not. So we have to study this absorption nature based on the actives and also formulations. So sometimes your active is retained strongly on your formulation. They don't want to release. So there's not absorbing. So here the science is you want to keep your active or the molecule in the formulation, but when the formulation is put on the skin, you want the active loves the skin and go into the skin. So that's the fast absorption means.
Yeah, I see. So it has so it means that it not always related to the size of the particle, but in terms of the hydrophilicity and the hydrophilicity of the active ingredients inside the product itself towards our skin. Okay.
Yes, it's
So
true.
I think that's the end of our that's the last question and the end. We are wrapping up the this webinar. Thank you once again, Dr. Nazmul, for your sharing your valuable insights and experience with us today. Today's session has truly highlighted how analytical scientists play a vital role in the product development pipeline, ensuring safety, quality, and innovation from lab scale research to to consumer-ready products. On behalf of IBD UTM, I would like also to thank all participants for your active engagement and continued support for our IBD webinar series. We hope today's session has been both informative and inspiring. Please stay connected with us for the upcoming webinars under the theme Biotechnology, Science, Business, and Society. With that, we officially conclude today's session. Thank you, stay safe, and have a pleasant day ahead. Wassalamualaikum warahmatullahi wabarakatuh. Bye bye. Thank you, Dr. Nazmul.
Thank you, everybody.